Hi, everyone. My name is Julia York. I'm a postdoc at the University of Illinois, and today I'm talking to you about my work looking at molecular temperature sensors in Antarctic fishes, specifically in the Notothenioid fishes, the largest group of fish that live in the Southern Ocean. So the best studied molecular temperature sensors are transient receptor potential or TRP channels, which are these proteins that insert into the membrane of sensory nerve cells and open in response to a heat stimulus or temperature stimulus. In this case, they allow positive ions to flow from the outside of the cell into the cell that depolarizes the sensory nerve and sends a signal to the brain. These channels are polymodal, so they can be activated not only by temperature changes, but also by ligands, changes in pH. Some are mechanosensitive and some are voltage sensitive. And this makes them key molecular integrators of climate change stressors. So fish have three main stressors caused by climate change. That's increases in temperatures, increases in oxidants, and decreases in pH. And so these channels are really on the front lines of interpreting those environmental signals into physiological signals in fishes. But we really don't have any understanding of how these channels might work or whether they might work at temperatures relevant for the Southern Ocean, which is generally between -2 and up to at the very maximum +4 degrees Celsius. So when I started looking at these channels in this group of fish, I found specifically one channel, TRPA1b, which showed evidence of up to 4 independent duplications, specifically within the Notothenioid clade. And so I thought this was a great candidate for potential characterization. The way I characterize these channels is by synthesizing the RNA of the channel in vitro and then injecting it into frog oocytes. Waiting a couple of days, the frog oocyte expresses the channel and inserts it into the membrane. And then I can measure the responses of the channel to heat stimuli that I control and measure using this thermistor, which is just downstream of the oocyte, in this electrophysiological 2-electrode voltage setup where I measure the current responses from the temperature stimuli. And so these are what the data look like. This red trace is the temperature trace and the black is the current trace. And I bring the temperature between about 5 degrees to about 40 degrees Celsius repeatedly and measure this inward current, so the positive ions flowing into the cell in response to the increase in temperature in the TRPA1b channel in cells injected with the TRPA1b channel, but not with water-injected cells. And so another way to look at these data are to plot the current versus the temperature with the time here in the color. So the darker curves are the earlier curves and the lighter curves are later. And you can see that as temperature increases, there's a linear section of the curve, and then at a certain temperature, which we call the threshold temperature, the current increases non-linearly with changes in temperature for the injected cells. And for reference, the blank cells only show this linear relationship between current and temperature. But you can see here that this threshold temperature is around 23 degrees Celsius, and that is way higher than expected for Notothenioids, which probably haven't seen these temperatures in 15 million years. So I tried a lot of different things to shift the threshold of the channel under the hypothesis that the cellular conditions in the native cell might shift the activation threshold. And I found that some of them, which are these lighter points here, did not shift the threshold significantly compared to the control. I'm plotting here the threshold versus the Q10, which is the temperature sensitivity. But I found several conditions that did significantly reduce the threshold of activation, and some of which maintained the temperature sensitivity of the channel. And then this brown one is the additive effects of methyl beta cyclodextrin and hydrogen peroxide, so an oxidant, and this compound which reduces cholesterol. But when I added cholesterol, which is this point here, I saw no shift. And so I think this effect of the methyl beta cyclodextrin is due specifically to disruptions in lipid rafts, which are associated with TRP channels. And notothenioids are specifically depleted in sphingolipid genes, which are associated with lipid rafts. And so I think that this change in lipid rafts may be a mechanism for the notothenioids to shift the activity of their channel to be at ecologically relevant temperatures. So thank you so much for listening. I have lots of people to think, and I will take any questions.